This is paper 6 from latest session, 2024. They're using a special indicator. It's called methyl blue. They told you all information about it here. They said that this indicator, when anaerobic respiration happens, it would change its color from blue to colorless. So you have one test tube. You just have yeast in water. Here I have some sugar with the yeast, little concentration. And here I have twice as much concentration. And I've done this in a water bath, so we've controlled temperature. They said we control the temperature at 40, and they incubated those test tubes for 3 minutes. Again, all of this is just being given to you inside the text, okay? So next, I have the readings. So you see, when I had that test tube where there was no sugar, no glucose, zero glucose, 10 minutes. Apparently, there was no respiration happening here. Here the color changed faster, and here the color changed the fastest. So again, when I had a lot of glucose, the indicator color, that methyl blue color, has changed or disappeared the fastest. So what do they want me to do now? They wanted me to convert the timings, the concentrations here, into a table of results. So usually in any table, when you're making a table, just keep in mind that in the table, you'll have to have at least 2 columns, okay? The left column is going to be for your independent variable, and the right column is going to be for whatever you're measuring, which is the dependent variable. So my case here, what's my independent? My independent was the concentration. So I would write the concentrations here, 3 different concentrations, and the dependent would be the time it took for this reaction. They wanted me just to convert the time here into seconds. So for instance, this would be 10 minutes. So I say 10 times 60. This would be 6 minutes. So I say 6 times 60 plus 2, right? Because 6 minutes is 60 seconds plus 2. And the last one, I have 2 minutes here. So it's going to be like what? It's going to be like 2 times 60 plus 12. And I'm going to record these inside my table. So one more time. Here we go. So this is my independent concentration. And here's my dependent after I converted all of my results into seconds. Next part, state a conclusion. What would be like the most obvious conclusion that you can come up with here in this investigation? It seems very obvious that when they have more glucose, there's like a negative correlation here, right? When there is more glucose, the time is less. It's getting shorter with time. So one more time, I say the rate of respiration is getting faster, really, as the concentration of glucose increases. Or you can say that time it takes for the color to change is faster when the concentration of glucose is higher. So you can write this, try to make it simple, basic. You don't need to go into any further detail about glucose because it's just worth 1 mark. What's the independent variable? Again, what does independent variable mean? Independent variable is the one you change. So in any experiment, the independent is the one that I change, and the dependent is what I measure, okay? So one more time, what did we change here? We were changing the concentration. What variable was kept constant? What did we control? So go back to the first page, check the numbers. Always, when I am being asked like what's being controlled, I check the numbers. If there is a number and there's a unit after, that should be a control variable. So for instance, total volume, it was recorded there in the first page and it was controlled. That 3 minutes, remember, and that 40 degrees Celsius, all has been controlled. So again, it's quite simple. You don't need to read the question more than once. Scan and find numbers. If there's a number and a unit after, then that must be something that's been controlled. Why do we stir? Why did we have to stir? Like while they're mixing the sugar and the yeast, they were stirring the content. They were stirring just to make sure that the sugar and the yeast, they were colliding, right? In any chemical reaction, you want the content to collide, the enzyme and substrate. Plus, you don't want the content to settle. If I don't stir my content, all of my yeasts will sink at the bottom because they're dense, right? So that's why I need it so can have the yeast distributed evenly all over the solution. So just how could you improve such an investigation? Improvements fall into 3 types. Reliability, mean you repeat. Validity means you control variables that they did not control, for instance. Accuracy, you use better equipment. So I'm gonna write the first and the last here. So I'm gonna say reliability, I say they should have repeated it, right? So 5 times at each concentration because they've already done it once there, right? Plus, to judge colors, I can use like a device called colorimeter, right? Colorimeter, very accurate in judging colors. Because if I was just relying on my eyes, it's gonna be very difficult to judge colors. Again, the colorimeter works like this. You take the solution, you insert it into the device, and it tells you a numerical value, rather than just relying on your eyes to judge the color, okay? So I'm gonna write the colorimeter here as my second improvement. Good. They also wanted you to do like a small calculation here. They said like, if you are to prepare 0.25 glucose from 0.5 glucose, how are you meant to prepare? So again, total volume here that I'm planning to prepare is 5, okay? 5 cm cubed. And I want the concentration to drop by half, right? I want half of the concentration that I have here in the original solution. So look at my plan. I'll take from that 5, I'm gonna split that 5 into 2. 2.5 here and 2.5 here. The first 2.5 is gonna be like the sugar, that 0.5 sugar. And if I want to dilute it by half, I'm gonna dissolve it into water. So put distilled water. That means that this 0.5 here will be halved, be half of what it should be. So once again, dissolve that 0.5 into water, distilled water, and you'll end up with what? You'll end up with half the concentration. Okay? Next, describe methods you would use to test the solution for the presence of glucose. This means they wanted you to use the Benedict's, right? Test of reducing sugars. And if the result is positive, then what color change you would expect? You would expect that blue color of the reagent to change to green, yellow, orange, or red, depending on the concentration. So here I'm saying that you could have used Benedict's to test for the presence of sugar. Benedict's solution. Don't forget in Benedict's that there's always like heating part, right? You need to heat because the test would never work unless there is heating. And usually like here is not necessary to mention the temperature, but if you have like more marks than those two, I would have mentioned that the water bath should be at 80 degrees Celsius. All right. Next, describe suitable apparatus to collect and measure volume of gases. Usually the best to measure volume of gas is gas syringe, right? Very accurate, and you don't need much effort. So just like put a gas syringe, collect the carbon dioxide, read, and you have the measurement of volume, okay? Next, they gave me here like 3 experiments, and they're measuring the volume of carbon dioxide being produced by respiration. So they said one of these results is an anomalous result. What is an anomalous result? Simply is an error. So which one of these results appears to be different or an outlier from the other results? It's clearly the middle one here, right? Experiment 2. Why is it like an outlier? Why is it an anomalous? Because it's very different from the other 2 results. And whoever was calculating the mean, he ignored it, right? He did not consider this result in his calculation, right? So I'm going to say it's an outlier, a different result, a result that doesn't fit with the other results, okay? Next, they said, describe how the student calculated the mean volume produced there. So as if someone's asking you like, how did he get that value? He ignored that, right? He took 1.8 plus 2, he added them up, 2.8, and then he divided by 2, okay? And he got that mean. So he eliminated, which is always the case. If you find that one of your results is wrong, then you must eliminate it from your average result. Here's his results. Again, we have time versus what? Versus the volume of carbon dioxide at 2 different temperatures. It seems that 35, I'm getting more carbon dioxide. The rate of reaction, as usual, is faster near the optimum temperature. So they wanted me like to compare for 2 marks the volume of CO2 at the cool temperature and at the warm temperature. So again, first of all, look at this beginning here. At the cool temperature, there was no carbon dioxide produced for the first 10 minutes. You noticed? So that's what I'm going to write first. But at 35, the results were always higher. You notice that it's getting faster and faster, or there's more volume at any reading at 35. Also, if you look at the final results here, apparently the reaction is finished here. There's no more carbon dioxide produced at 25 and 35, while here it continued to increase. So again, in general, 35 is always faster. There's more carbon dioxide. And for that reason, the reaction finished here for 35 because I'm no longer getting any more carbon dioxide. Now they wanted me to do what? They wanted me to draw a curve, draw a graph for these two sets of results. So as always, when you're to draw a graph, the left column is your x-axis. So that is my x. And the right is your y-axis. Here I need to include both at 25 and at 35. So let's start first with what? Let's start first with the x-axis. So I'm gonna put those results here from 5 and 10, and I want to expand it as much as possible. So I'm gonna put them like in 5s here, 10, 15, 20, 25, and 30. That should be good enough. And I should never forget to write the headings here, right? So write time / minute. What about the y-axis? The y-axis is stretching from 0, the origin here, all the way to 3.2. So ideally, again, I want to expand my grid. I don't want like to end up somewhere here. So I would try to stretch it. So for instance, I could put every small box is worth 0.2, and I go by 0.4 here. And that should be good enough to expand it and reach to the top, all the way to the top, right? So that's going to be 2, 2.4, 2.8, and 3.2, okay? And again, I should never forget to label my y-axis. So the mean volume, I'm just going to copy that one here, a mean volume of carbon dioxide, and include what? Include the unit. So I'm going to write cm cubed, okay? Now I'm going to plot each one of those graphs and try to make a key. For instance, I can make a key here. One of the graphs is going to look like this, and one of the graphs looks like that. I tell them that this is for the 25 cm cubed. No, 25 degrees Celsius, I should say. And the other one for 35. And I plot according, okay? So again, I'll show you I've done this before because I can barely see the grid when I'm on the tablet here. So that's the one I've done earlier. You see what I've done? I plotted the values, I plotted the results. I can label it here at the end, see 25 Celsius, 35 Celsius here. And I always, in this subject, we always join with a ruler, right? So always join your results with a ruler. Whatever you plot, point to point, join together like the way I've shown here, okay? So once again, the left column is your independent variable, it's the x-axis. The right column is your dependent variable and is on the y-axis. Next, they said estimate the time taken to produce 3 cm cubed of carbon dioxide at 35 degrees Celsius. So let's find at 35 degrees Celsius how much carbon dioxide produced at 3 cm cubed. So the time, so I'm gonna take 3. 3 is here, right? So 3 should be halfway. I'm gonna draw like a horizontal line like this, and I'm gonna draw a vertical line all the way down, and I'm gonna read that from my x-axis, okay? So you read what's shown here. Again, ideally, so that should be like nearly 22, 23 minutes, okay? So again, I went to the 2 on the y-axis, drew a line like this, vertical line, and I showed my answer here at the bottom, okay? Next, carbon dioxide. You can test for carbon dioxide with lime water, but there's also another indicator called hydrogen carbonate indicator. The indicator is red, but if you bubble CO2, what color change you would expect to get? Usually, hydrogen carbonate indicator comes with 3 different colors. It could be red. That's where the pH is neutral, right? If the pH is alkaline, it's like, let's say 14, 9, high pH, it's purple. That's usually when the CO2 level is very low, right? But when CO2 levels are very high, you would expect what? You would expect the color to be yellow. That's when the pH is below 7, okay? And again, this reason it turns yellow here, because CO2 is acidic. CO2 is an acidic gas, right? You bubble that, it drops the pH, and the hydrogen carbonate indicator is sensitive to the change here and gives you that yellow color. And next comes the drawing. They gave me this picture here, and they said, draw a diagram labeled of the cell labeled X. So I'm gonna apply the usual rules that we always do whenever we are making a biological drawing, those SOPD. First of all, I'll make sure that my picture or the diagram I'm actually drawing here, I'll make sure that it's bigger, as big as the space I was provided, right? So again, size, number one, is my first priority. Then I'll show the outline. I'll show the picture as just an outline. And then I'll show that's the proportional size. So once again, let's start first with this size. I'm gonna draw that cell as big as the space I was provided here as an outline. I could show the wall because there's a wall of the neighboring cell there, like this, right? And see, these are not good here, so I could just get rid of them like this. It shows that's a continuous line, outline. Okay, now it's better. And then I'm gonna draw that large vacuole you have inside. That, see, it has like a small extension here, and there's one here, right? And all right, and there's a nucleus, so just put the nucleus where it should be. Don't shade the nucleus, that would be very wrong, right? So again, in any biological drawing, first mark usually, let me show you the mark scheme, would be better. So first mark, it's a clear outline, no shading, as big as the space. So if you expand it, you don't need to measure, don't waste time measuring, make it as big as the space you have on your exam paper. They gave you a mark for the detail, those extensions, do you remember those extensions I showed on the cytoplasm like this? And the position of the nucleus, okay? So that should be good enough. I could show you one I did earlier, which is slightly better. Okay, now they gave me the magnification too here, right? So they're gonna soon ask me to work out the actual size of that drawing, which is shown here. So here's my ruler. I'm just gonna measure the length of this line. Now this will really depend on the printer or the paper you're using here. So I'm just gonna measure it here. Let me just assume it's 10. Again, that's very inaccurate. I'm just assuming it's 10 centimeters, okay? Line PQ, measure that length on the cell. So with your ruler, grab your ruler and measure the line from P to Q, 10 centimeters or 100 millimeters, right? And then they said, calculate the actual length of the cell. So that's the original measurement, right? That's the line PQ. Now they said calculate the actual length of the cell X using the formula and your measurement. So again, I'll work out the actual size here. So again, the image was 100, right? The magnification was 340. So again, I'm gonna say 100 divided by 340. This should give you like nearly 0.3, okay? So that's the actual length I have there on that scale on the diagram, right? So it's 0.3. Final part, it's about osmosis here. They said water moves into a cell by osmosis. Plan investigation to determine the effect, that's like the most important line here, effect of concentration of sodium chloride on osmosis in a plant tissue, okay? So again, just any plant tissue of your choice. I could do it with onion, I could do it with potatoes. Regardless, let's just plan, because really this question is worth planning before you write, before you commit yourself to 6 marks. What am I gonna change? That's your independent variable. I'm gonna change this, right? I'm gonna change the concentration of salt, right? Concentration. The measurement is gonna be like, whatever I'm gonna measure here is, maybe I'll measure the mass, right? How much is the change of mass of potatoes or onions? Because when you soak them, let's say this is my potatoes. If I soak those potatoes in the solution, some of them will absorb water, will increase the mass. Some of them will lose water and decrease in mass. It doesn't have to be the mass. You may measure the length, change in length, for instance. What to control? As always, I can control temperature. I can control the origin of the like the plant tissue I'm extracting. And as always, I repeat 5 times and I write safety. Since I'm cutting potatoes, I wear gloves, for instance. So this means I have now a clear idea on what am I gonna do. That's my independent variable, like a series of different salt solutions. My dependent is gonna be like, I'm gonna cut some onions or potatoes. I'm gonna cut them into cylinders like the ones I have here. And I'm gonna soak them into these solutions. You see those solutions I have on top? I'm gonna soak them there, and I will find how much is that change in their mass. Ideally, you find the percentage change in mass. So let me show you that working out here, though, the procedure. So again, here is my plant tissue, the potato I'm planning to use here. I'm gonna cut cylinders like this, dry them first so that you lose the water on the surface. Try your best to make them same size, but it's not gonna be perfect. Then measure the initial mass. You see the initial mass is more or less the same. Next, I'm gonna soak them into solutions, each at different concentration. You really have to wait here, right? So wait realistically like minimum 6 hours. I will wait just like 2 days, just to be on the safe side. Some of them are gonna shrink. So some of these will shrink in mass and size, and some of them will expand depending on the concentration. So for instance, if I soak it in distilled water, it would gain water and increase in mass. If I soak it into highly concentrated solution, it would shrink and lose mass. That's not our case here. It's just like you need to write the procedure, okay? So again, I'm gonna prepare like 5 different solutions, all the same volume, right? Different concentration. Cut cylinders from the same onion, potato, same size. Measure the initial mass, because I'm planning to see the percentage change in mass here. And then soak them, right? For how long? Hours, right? No minutes here, matter of hours. And then calculate what? Calculate the change in mass, percentage change in mass. Don't forget to wear gloves for safety, because cutting may damage someone's fingers, all right? All right, so this means we're fully done with this topic. I'm gonna now let you go, unless you have questions.